So the next session will be Associate Professor Misty Jenkins. She's the laboratory head at Walter Eliza Hall Institute, and she'll be talking about targeting the immune system in brain cancer.
Thank you. Thanks, Maria. Thanks so much for the introduction, and thanks so much for having me here to come and speak to you today. Really what I want to do is I'm going to shift tack a little bit and talk about the preclinical work that happens before we get to the clinic. And because in order to take things through to the clinic, Jim's just really nicely outlined clinical trials and Liz's talk as well. This will build on that. But at the start of that pipeline and tell you what sort of goes into how we can bring new things to the clinic in the first place, which is where we want to get to. So I'm from the WEHI, the Walter Eliza Hall Institute of Medical Research, and this is one of Australia's oldest medical research facility that sits just behind the Royal Melbourne Hospital. And I'm a cancer immunologist. For those that don't know me, I'm here to talk about T cells. Now, we all have T cells zooming around our body through our blood and our lymph and our tissue and in our brain. And this is just a beautiful microscopy image of a real living T cell crawling across the dish. And in the movie on the right-hand side, you can see the T cell attaching to a cancer target. And so the job of T cells in our body is to zoom around our body and find malignant, infected, transformed, dangerous cells, and ultimately deliver the kiss of death and take them out. So this is happening in our body all the time. And so the lethal hit towards the target is directed by this T cell shown here in red, directs this little red dot, the centrosome, sort of like the laser-like definition, and then onto the target here, and this blue target you'll see blow up, and these T cells deliver a payload of toxic granules as shown here in red, which kill the target. Now, when we're designing living T cell therapies, of course, one T cell and one target doesn't make for a very efficient immune response, and we want one T cell to be able to take out loads of targets, and that's just great because they are serial killers. So these T cells, you'll quickly see, deliver the kiss of death to 1, 2, 3, 4, 5 targets in very quick succession. So they're quite efficient when they're doing their job properly, and they're able to go around and kill the cancer cells. And so in recent years, we've been looking at using these and manipulating them to use as a living therapy. Now, sometimes, of course, the cancer cells are invisible to the immune system. And so that's what happens here. This is a T cell here trying to kill this cell, and it's just not dying. And because the T cell isn't able to deliver the right kiss of death. And so what happens here is then sometimes you can get a build-up of inflammation. Or it's just completely invisible to the immune system. So, having cancer and not having cancer is sometimes the difference between that war and when the immune system is losing the war and the cancer is invisible to your own immune system, so some drugs, you'll hear about, like checkpoint markers, are drugs which enable to sort of wake up a sleepy immune system, because a lot of tumours will send your immune system to sleep and sort of make it invisible to it. And so that's great if you already have that pre-existing immune response to your tumour, and some patients do. But in the case of brain cancer, this is a bit more tricky, and a lot of patients don't have it. But also then what we can do is we can create it. We can actually engineer it from a patient's own immune system. And so this is what I'm going to talk to you about today, chimeric antigen receptor, or CAR T cells. So we're essentially taking patients' own T cells and beefing them up and making them into a Mr. T cell, which is a cultural reference point lost on the younger members of the audience. But those of you familiar with the A team, that's really what we're doing. So we're creating a living drug. Now, when we think about cancer therapy and cancer treatment, it's really been founded on these 3 pillars, and Jim has already really nicely outlined this. So we've been using surgery for centuries, actually, dating back to the ancient Egyptians. Of course, that's much more sophisticated now, as Jim's outlined. Radiation around the 1900s, and then, of course, chemotherapy, and you'll all be familiar with the Stupp protocol for brain cancer. And so immunotherapy has formed this 4th pillar of cancer treatment and the way that we can treat tumours using the patient's immune system. Now, this is the most sciencey slide that I have, but it's essentially, if you sort of look at this line here across the middle, this is the membrane or the outside of the T cell. And what we're doing is we're able to give the patient this receptor. So this is sort of the key that's going to fit the lock of the tumour cell. And so this outside binding part is like the magnet. That's the key that's going to bind to the cancer cell. And then we can engineer this receptor with a whole bunch of these important signalling molecules that are normally found in a signalling T cell receptor, which is basically the kill signal. And this basically redirects the T cell to kill the cancer cell. And so this sort of approach has been used with great success in blood cancers. So this is really the first paediatric patient that was treated on Earth, Emily Whitehead. So you can see with these dates here how recent and cutting edge these therapies still really are. She was treated for a very aggressive and refractory leukemia, and she's now, as of this year, still nine years completely cancer-free and has been cured of her cancer. So the challenge for us is, how can we take those success stories like Emily and translate them into the brain cancer space? And these sort of facts and figures are not lost on this audience, I know, and because new treatments are urgently required, both in the adult and also the pediatric setting for tumours like DIPG or diffuse midline gliomas that grow in the brain stem and are surgically inoperable, because this is a part of the brain that controls your breathing and your swallowing. So they cannot be surgically removed. And so, we need ways in which we can target these tumours specifically, but leave the healthy brain cells unharmed. And that's sort of really the power of this immune response. Similarly, for tumours like glioblastoma, which we know is one of the most common tumours, but with the poorest survival. And so this is really just a simple schematic of what this process looks like in the clinic. Excuse me. And so after diagnosis, blood is taken from the patient, where their T cells then are isolated from the blood. They're then grown up in a dish, and they're given that all-important CAR, that killing receptor that's introduced to the cells. And then the cells are grown up in the lab, and then they're infused back into the patient, usually after some lymphoablations that sort of make space for the new T cells. And then they're able to be delivered either through in the vein or straight into the brain, and go in and kill the tumours. And so this is what we've been doing at WEHI, is then to build this pipeline so that we can rapidly now take these sort of approaches and build a pipeline to deliver this sort of personalised medicine for brain tumour patients. Now, this involves first knowing what to target on the brain tumour cell. So what is the right key that's going to fit the lock? What is different about that tumour cell that's not going to kill a healthy cell? And so this is a really, in my mind, one of the most critically important pieces of the puzzle that we need to solve. And so we're doing this in my lab at WEHI. Once we identify what those targets are, we can then make those engineered receptors, and then we go into the lab and do a bunch of functional experiments to work out how well they can kill their targets, and then we can test them in mouse model. And we use mouse models for this, and this enables us to then look inside the tumour environment, look how the T cells are trafficking in, and how well they can kill those brain tumours, and then with the plan to take that then into the clinic and be able to translate these into people. And so we did this with a very already well characterised target called EGFR-V3. And about 40% of adult glioblastomas have this mutation. And so by making our CAR T cells against this target, you can see here these little purple dots are brain tumours in mice, and that we're able to completely clear and completely cure these mice of their brain tumours. And I'm proud to say that these cohorts still, a year later, still running around on the shelf, very happy. And very much alive. So that was really quite exciting for us, to show that we could make these therapies from scratch and that they were working. And so now the next question is, so how can we now put more tools in our toolkit? We need more keys to fit the locks. We need to know what are other targets that are present on the surface of these brain tumours we can target. And to date, this is something that hasn't had a lot of research in it, which is sort of where I'm interested in participating in this space, because particularly in brain tumours, there's been only a handful of targets identified, whereas there's been a lot of success in the blood cancer space. And so this is really where we think we can make a big impact. And in addition to that, we're also then looking at the interplay of how these cells get into the brain and how we can stop them getting made to be sleepy by the tumour. The tumour can shut down. It can be very immunosuppressive environments and shut down the immune response in the T cells. So to do this, we can use these little mice here, and we can actually, they recover very well from this surgery, and we can watch the tumours grow, and then we can give our therapy and then really watch those tumours melt away and look at the interactions of those T cells and all of those other immune cells too. So this is a movie. We can take these longitudinally over a number of weeks. And so these little yellow cells you can see here are the T cells infiltrating, and then this big pink mass here is the tumour. And so what we do is we take a very fancy microscope, and this is in the living animal. This is in the living mouse. We can see single cell resolution of these yellow cells coming in and infiltrating and getting into that tumour, and we can see it kill the tumour cells. So this is the first time this has been applied in Australia, this technology. In fact, this is the only platform established to do this in the Southern Hemisphere. And we have this at WEHI, and it's called a 2-photon microscope, and using this to look at these little intracranial windows. And this is telling us a lot about the tumours. This is the vasculature and the blood in the brain, and you can see this pink tumour here growing in this mouse. And we can then look at a depth down with the microscope. It can go quite deep into the brain, and we're able to then really map what that really looks like. And so this is a really powerful platform because it enables us to then look at this environment and the effect of all kinds of different drugs. So not just T cells, of course, but lots of other therapies as well. And then we can also look at the role of these T cells with these, what's shown as microglia here in green. So these T cells are in yellow. The tumour is in red here. You can see those little T cells moving around. And then we can also see them interact with the green cells, which are the immunosuppressive cells, which shut down the T cells. And so by understanding those interactions too, we can then use other drugs in combination to then say give T cells, but also then to shut down and to really dampen down that suppressive immune response so that we can get the best robust immune response we can to attack these brain cancer cells. So CAR T cell therapy is a really important piece of the puzzle. So, currently standard treatment is chemotherapy, radiation and surgery. But I think, by using CAR T cells, both with single receptors, and I should say, the field is getting quite fancy with its genetic engineering in terms of targeting multiple proteins at once. And also being able to then get the T cells to locally secrete therapeutics right into the brain. And so that, of course, helps with toxicity because you're getting the effects right at the tumour site and not systemically throughout the body like you do with chemotherapy and other drugs. And then we can also use this sort of CAR T cell therapy approach in combination with other biologics such as antibodies, called checkpoint inhibitors and other small molecules and drugs. And there was a CAR T plus Pembro trial completed earlier this year. And those are yet to be reported, but I think there certainly is some hope here. And I just want to point out that one more thing before I finish, which is in terms of all other solid tumours, brain cancer is actually the one indication where there has been good tumour regression in a patient treated with CAR T cells. And that's the only real response in solid tumours that we've seen so far on planet Earth, and it's been in brain cancer. So I think brain cancer really is leading the way in this area. And I certainly think there's a lot more that we can do. And I'll just finish with this slide, which is, I think that sort of certainly here in Melbourne, there really is high potential for translation where in order to make these T cell therapies, they have to happen in a very highly regulated specialized facilities called clean rooms, and they have these at the VCCC with a company called Cell Therapies, and we're all connected by bridges, there in Parkville. So it's very easy for us to have these therapies made now and to be able to translate them into the clinic. So I think there's a really huge opportunity to translate into the kinds of clinical trials that Liz and Jim have already nicely outlined this morning. So with that, I'll just thank my large team who contribute to all of this preclinical work, and none of this could happen without the important collaborators, funding, but also patients and their families. I should point out that, in order for us to really find new targets on the brain tumours, it does rely on accessing fresh tissue. And so, donated tissue from biopsies or surgeries, when you have a tumour removed, You know, instead of it going into the bin, it could actually go into a lab, and that's how we actually find new targets and enable us to really help patients. So I just want to thank those patients for those generous tissue donations, because they're absolutely critical to the work. Thank you.